Oxidative stress is a major driver of both aging and cancer, arising when the production of reactive oxygen species, ROS, overwhelms the body's ability to neutralize them. These highly reactive molecules, mainly generated by mitochondria during energy production, can damage DNA, proteins, and lipids, alterations that accumulate over time and contribute to cellular dysfunction, senescence, and malignant transformation. Autophagy plays a central role in counteracting this process by acting as an internal detoxification system that prevents the buildup of oxidative damage at the cellular level. When autophagy is functioning efficiently, it selectively removes damaged mitochondria through a specialized form known as mitophagy. This not only prevents further ROS production from faulty mitochondrial complexes, but also supports cellular energy balance by ensuring that only healthy organelles remain active. In doing so, autophagy interrupts a vicious cycle in which damaged mitochondria generate excess ROS, leading to more damage and greater mitochondrial dysfunction. This cycle, if left unchecked, accelerates aging and increases the risk of mutations that drive cancer. Moreover, autophagy clears oxidized proteins and lipid peroxides, which are not just byproducts of stress, but also contributors to cellular senescence. Senescent cells often accumulate as organisms age and are characterized by a permanent growth arrest and the secretion of pro-inflammatory cytokines, growth factors, and matrix-degrading enzymes, the so-called senescence-associated secretory phenotype, SASP. This inflammatory milieu does not remain confined to the individual senescent cells, but alters the surrounding tissue environment, promoting further damage, immune dysregulation, and in some cases, the stimulation of tumor growth in nearby pre-malignant cells. By preventing or delaying the onset of senescence through the clearance of oxidative lesions, autophagy indirectly suppresses these downstream effects. However, the protective role of autophagy in mitigating oxidative stress is not unlimited. With aging, autophagic activity naturally declines, leading to inefficient clearance of damaged cellular components. As ROS levels rise, cells begin to experience cumulative oxidative damage, which not only accelerates aging-related degeneration but also sets the stage for DNA mutations and epigenetic alterations that can drive tumorigenesis. In this way, impaired autophagy contributes to a dual burden. It weakens the cell's ability to fight oxidative stress and simultaneously permits the cellular conditions under which cancers may arise. Therapeutic strategies that seek to restore or enhance autophagy, whether through fasting, pharmacological agents like spermidine or rapamycin, or genetic modulation, show promise in reducing oxidative damage and extending cellular health span. These interventions may offer dual benefits, preventing senescence and maintaining mitochondrial function, which are both central to longevity and cancer prevention. Nevertheless, they also carry potential risks, especially if applied in the context of established tumors. As with other forms of metabolic stress, autophagy may protect cancer cells in harsh conditions, including during chemotherapy, by helping them clear oxidative insults and maintain viability. Thus, while enhancing autophagy appears beneficial in the context of aging and early disease prevention, its application in clinical settings, particularly in individuals with cancer or high cancer risk, must be approached with caution. Autophagy's role in managing oxidative stress is protective in healthy tissue, but once a malignant process has begun, the same pathways may inadvertently support tumor survival under therapeutic assault. The relationship between oxidative stress, autophagy, and cellular senescence is a cornerstone of our understanding of the biological aging process. By regulating the quality and function of mitochondria and clearing oxidized macromolecules, autophagy acts as a molecular defense system that helps preserve cellular integrity across the lifespan. When this system falters, the floodgates of damage open, not just increasing the pace of aging, but laying the biochemical groundwork for cancer to emerge. Supporting autophagy, then, is not about forcing the system into overdrive, but about keeping it responsive and balanced, a quiet but essential force that keeps oxidative chaos at bay.